So here's the hypothesis that surrounds how contactin 2 can mediate multiple sclerosis and potentially be a causative agent of multiple sclerosis, sort of. The idea is that someone who is susceptible, and that could mean a lot of different things, but perhaps a deficient immune system, some sort of pre-existing condition, right? But someone who's maybe more susceptible could get infected with a minor viral illness. So someone who is susceptible gets a minor virus. This illness, right, this viral illness leads to expression of a molecule that looks similar to contactin 2. So the illness from the virus. If you know anything about viral life cycles, right, is that they hijack cells and cause the cells to produce their own proteins. Well, the illness from the virus is in part due to production of a viral protein that looks a lot like contactin 2 structurally. And then when the immune system responds, I think you can see where this is going, but the immune system is unable to differentiate between self and viral protein. So the immune system can't differentiate. Self and not self. And so the immune system sort of overreacts and attacks anything that looks like contactin 2 and contactin 2 itself. A second hypothesis with respect to the cause of this disease, I'm going to erase this, has to do with a persistent infection, right? If that was hypothesis 1, the second hypothesis with respect to cause is that if there's a persistent, potentially viral infection. And that this persistent infection triggers long-term activation of the immune system. Right, and so this long-term activation leads to an ongoing damage to myelin, right? Because this immune system will continue to fight the persistent infection, that loss of myelin, and the associated oligodendrocytes is ongoing. There is precedent for this. There's a virus called TSP, tropical spastic paraparesis. Tropical spastic paraparesis. And the symptoms of this tropical spastic paraparesis, which is a virally mediated illness, is weakness in the legs, loss of bladder control. And this looks a lot like multiple sclerosis, these symptoms. So this resembles MS, and it's caused by a virus. Okay, it's caused by a retrovirus. So this retrovirus is sort of at continual war with the body, and this continual immune response leads to these symptoms, right, that we just mentioned. And so something similar, hypothetically, could be going on with MS, though evidence for this and evidence for any of these has been really hard to come by. Okay. So the diagram to the right indicates some disease progression, and we'll talk more about the immune-mediated signals in a minute here. But what I want to give you an idea of is the progression and how this can work in phases, right? So initially, what's commonly seen is that in the first phase, right, where the first motor symptoms will be noticed, that's due to a loss in myelin like you might expect. So progression. Right. The first thing to note is early symptoms appear as the myelin gets lost. So the immune response would either break down the myelin and or kill off the oligodendrocytes. Okay, but oftentimes patients who have some symptoms recover. And so what you can see here, right, so here's the big macrophage that's working to break down some of the myelin. And you know, I think, that under the myelin sheath, there aren't very many voltage-gated sodium channels, right? Those voltage-gated sodium channels exist here, right, at the nodes of Ranvier. And they're important in sort of boosting the signal such that it can propagate all the way down this axon, okay? And so without myelin, right, if this myelin gets degenerated, that signal, right, might get boosted here, but tends to run out of strength and may result in a failure and not get all the way to the axon terminals where we release neurotransmitter, right? So the importance of the sodium channels here at the nodes of Ranvier is to boost the signal here and here and here and so on. And the importance of the myelin is to insulate the signal such that it stays above threshold. And once we get to another node, right, so as we go from this node to this node, as long as that action potential stays above thresholds, once it reaches this node, voltage-gated sodium channels open up and that signal gets boosted again and we go to the next node. Okay, that's saltatory conduction. And because myelin is lost, this results in a failure of saltatory conduction. Now, like I said though, oftentimes people feel as if they recover from this disease, and because even though you can see in this middle image, a lot of the myelin is gone from here. What happens is that those sodium channels that typically only exist at the nodes have been upregulated and expressed throughout the axon now, right? So these sodium channels, these sodium channels are present throughout the bare axon. But an upregulation in sodium channels seems to reduce the symptoms because now the signal doesn't need to be boosted. It can just travel down the axon and open up sodium channels throughout the length of the axon, right? So upregulation in sodium channels that are now expressed throughout the axon. This word is supposed to be throughout. Right, when this happens, the patient usually feels as if they're recovering. the symptoms tend to lessen, right? Because now the signal can be propagated down the axon again. But over time, right, as this is degenerative, you can see what happens is that the axon degenerates, right? So that's why we talk about MS as not only a demyelinating disease, but an axonal degeneration disease. Okay, and now we see a new phase of this disease, a more severe phase of this disease, because now there's no way for that signal to reach the postsynaptic cell here, right? And you can see that this is not healthy for the postsynaptic cell either, right? There's a loss of trophic support to this cell. One of the things that we also think is important in this degeneration is a rise in intracellular calcium. That is, excessive calcium helps mediate this, right? So you can see that calcium channels have been expressed now. And we think that that overexcitedness, right, too much calcium leads to toxicity and helps to degenerate or degrade the axon. Okay, so the result of this is really a complete loss of signal propagation. All right, let's take a look at the immune system and how the immune system responds to this disease. And so we're outside the blood-brain barrier out here, right? And everything inside, right, this is within the blood-brain barrier. So make that note, and now let's put a heading up here, which is going to be immune response. So initially, these CD4 positive T cells, right, that are these blue cells right here. So CD4 is just a protein that marks them and gives them some identity. So CD4 just means that they're a population of immune cells, right? It's a particular type of T cells. So these T cells of the immune system they encounter macrophages or either other dendritic cells. This is kind of a strange term, but in this case, dendritic cells aren't dendrites. They're a type of immune cells. So, but they encounter these cells and they present the antigen that the immune system will want to attack, right? So these T cells encounter other immune cells right, that present the particular antigen of interest, okay? And so you can see here, right, this T cell is interacting with one of these dendritic cells that's presenting the antigen. However, these CD4 positive T cells don't do much beyond that, besides releasing specific chemicals, cytokines and chemokines, which trigger other immune cells to the scene. So these CD4 cells, once presented with the antigen, oops, this is supposed to be release. Cytokines, chemokines, right? That's right here. And what this does, these cells are these molecules, cytokines and chemokines, are basically attractants for the immune system. Okay, so these chemical signals help attract other immune cells. Macrophages a population of cells called CD8 positive T cells. B cells. Right, so these B cells here, and these macrophages here, right, are responding to the production of the inflammation, that inflammatory cytokine or that inflammatory cytokine, chemokine that was produced by the CD4 cells, okay? And now that the macrophages and other cells, CD8 cells, are on the scene, right, they can recognize these proteins on the oligodendrocytes themselves, or on the myelin themselves, okay? And so you get demyelination and axonal degeneration because of this. The other important thing to mention, right, and then the video will be done for this sort of category, or this disease, is that afterwards, after the oligodendrocytes break down, is a characteristic glial scar that forms. And what this prevents is remyelination, right? So you can imagine that this wall of glia tend to cordon off the site of injury, try to protect, right? So here's a glial scar, try to protect other oligodendrocytes from meeting the same fate, okay? The problem is now the axon can't regrow, the myelin can't come back, the oligodendrocyte can't remyelinate. So what we think of as a protective mechanism, the formation of this glial scar, really prevent regeneration opportunities, okay? So there are ways that people think about treating this, with maybe with stem cells, but I don't think that we have any great treatments yet right now. So again, just by summary, the immune response is that CD4, these CD4 T cells, recognize the antigens that are presented to them by other immune cells, and they dump out cytokines, right, and chemokines. That works as a chemoattractant to bring other inflammatory cells onto the scene, and those inflammatory cells can recognize whatever it is that the myelin contains, that the oligodendrocytes contain, right, as a target to cause breakdown of the myelin and destruction of the oligodendrocyte. Okay, good. Went a little long, but maybe we'll try to keep today's set of videos to 4 instead of 5. And I'll come back and we'll talk about ALS next.